Recording. So, to be honest with you, this isn't something that I'm engaged in or all that interested in unless it put us out of business in making immunotherapy. That's what it comes down to, is the conditions for making immunotherapy. I know the one person in the room certainly might talk about this would be Frank, since he manages the lab over at Northwest Asthma. Do you have comments?
Yeah, basically, I think everyone's take on it is that there's somebody new at the relevant government agency that says, well, let's go after everybody. That's not a pharmaceutical company that's like providing something to a pharmacy that is unadulterated. And it's essentially there's requiring that you have basically a hazmat suit and a hood with certain flow rates and so forth. And the rejoinder, I know there's been countless letters from the leadership at the college and the academy of saying, here's 80 years history of, there's no case reports of there ever being a problem. It's a little different than somebody smashing up tablets at a pharmacy and doing this and that and the other thing. So it remains to be seen where this is going to go. But, I think we could pull it off in our office, but I think a lot of places that mix antigen probably couldn't without thousands of dollars of, it would be almost a money loser, obviously. So it is a big deal.
One of my friends is a pharmacist who's trying to decide whether she wants to get into sterile compounding. So I've heard this whole story from the pharmacy side. And this is all still fallout from that fungally contaminated intraspinal steroids from Massachusetts, where they got all those people sick. So Pharmacy Law 797 now says that if you open something, you have to dispose of it within 30 days of any bottled product. And they're trying to enforce that all the way across the industry, which is coming to us now. And she figured out that you have to have a negative pressure unit inside a positive pressure unit inside another negative pressure unit to currently meet the state's requirements for sterile compounding. And she says it'll cost her at least $60,000. So she probably is not going to do it. But it's all fallout from that fungally contaminated steroid issue.
So that would mean we'd have to throw away antigen bottles monthly and reformulate them?
Or they'd have to package them in 1 month aliquots. So you get 12 bottles for 12 maintenance shots instead of 1 bottle for 12 maintenance shots. Because the closed bottles you can keep at whatever the experimental standard is, but the open bottles it's 30 days and you throw them out.
So I know from just a little bit I looked at, I guess they got like 8,000 responses from the members of the academy and the college, which at least pushed this down the road. It's not a definitive decision, but Art is, I don't know if he's listening, but is always interested and involved in kind of legislative issues so it's important to watch this, and if it rises again to threaten our practices, we definitely need to respond to it. So I don't know if there's anybody else. Bill, this wouldn't be an issue. The university, I'm sure, could meet these standards, but
yeah, because they use actually trying to become a specialty pharmacy, so I would imagine they would meet every criteria here. Ours are made out in-house by inpatient pharmacy.
Okay. Any other comments on this? Otherwise, we'll move into the articles. Drew, you want to get started first with your article from the New England Journal, and Teal is here. I think he's...
Teal did a critical review of this, so I'd be interested to get his thoughts, and he can correct me many times I'm wrong about this. All right. Actually, before we begin, I bring this up every year. All of the talks from the past are saved on our website. Most of you know this, but if you go to the Allergy and Immunology Fellowship website, come down here and go to A&I Teaching Series. You can access any of the past talks that we've done. They're all on YouTube now. And I review some of these when I give talks. They're great resources. And I know that one. I picked a weird one, but they're great resources if you guys are at all interested.
Just a comment, I've actually had patients, even watch some of them, particularly patients with hereditary angioedema. Years ago we had Mike Frank here, who gave us just a brilliant talk, and I've had patients with the diagnosis watch his presentation. So that's another use for some of these.
All right, so no disclosures. So I chose to review a recent article that came out in the New England Journal from August 4th. There's a pointer. Innate immunity and asthma risk in Amish and Hutterite farm children, which I thought was a really interesting article. Kind of the pretext for this, there's some people in the same group put out a paper in 2002, which I think many of you know, environmental exposure to endotoxin and its relation to asthma in school-aged children. And it looked at children aged 6 to 13 in a huge cohort in Central Europe, Germany, Austria, Switzerland. And there were both farming and non-farming households in this area, and they looked at levels of endotoxin in the bedding in relation to clinical findings and then cytokine production profiles. They found that endotoxin levels were inversely related to the occurrence of atopic disease, allergic rhinitis, atopic asthma, atopic sensitization in general. In other words, the more endotoxin you have, the less risk you are to develop atopic disease. Cytokine production by leukocytes, including TNF-alpha, interferon gamma, IL-10, were inversely related to endotoxin as well. Again, the more endotoxin you have, the less cytokine production that you have. And their conclusion from that study was that environmental exposure to endotoxin may have a crucial role in the development of tolerance to ubiquitous allergens. So this is kind of what got this field going, and there's been a lot of other research in the interim, but I thought this was another interesting study to add to this. So, background, there, as we all know in this room, been a dramatic increase in the prevalence of allergic conditions in the past 50 years or so. And we know that the environment plays a critical role in the development of the immune system, especially early in life. Again, they point to this study that I just reviewed saying there's significant protection from asthma and allergic disease in children raised on traditional dairy farms. In other words, the more animals they were exposed to early in life, the less their risk was to develop atopic disease. And so they chose these two very unique populations, the Amish and the Hutterites, which are both agricultural populations whose lifestyles are really similar, other than one aspect. The Amish are very traditional. They, one horse, they plow the fields themselves. They have the barns close to their homes, and they're just one-family farms. Whereas the Hutterites are a communal practice where they share in all these duties. The land is shared, and they have very nice farming equipment, very up to date. The Amish they studied were from Indiana, and the Hutterites were from South Dakota. The Hutterites are mainly located in the Midwest and Western Canada. There were striking disparities in the prevalence of asthma between these two populations, which is why they studied them. The Hutterites have almost fourfold increase in asthma compared with the Amish. So both groups, the reason they studied this, have been reproductively isolated since they got here in the 1700s, 1800s. And their lifestyles are very similar, other than that farming risk factor, which I talked about. They both have a lot of siblings. They have diets rich in fat, salt, and raw milk. They have low rates of childhood obesity. They have long duration of breastfeeding, minimal exposure to tobacco smoke and air pollution, and they have taboos against indoor pets. So the prevalence of asthma in school children in the Amish is around 5%, and this has been done in multiple studies. And the Hutterite is closer to 20%. So again, in most studies, it's a fourfold increase, so not subtle.
Drew, do they have similar vaccination rates for their childhood immunizations?
Yes. And that wasn't in the study, but I thought I read that in researching them.
And is it high or low? Do they stay?
What I read, it's high. Don't quote me on that, but I did see one study saying they do vaccinate.
Theo, do you know if that's an issue?
About the vaccination rate?
Yeah.
It wasn't in the study, but I saw it in one of the reviews I read on these populations.
I think one of the other things that I'm not sure you mentioned is generally thought that the genetic risk of asthma is similar in these two populations.
Yeah, they have a great genetic analysis of this. Yeah, very similar genetically.
So they're trying to really isolate the environmental component.
So the Amish, it's a traditionalist Christian church, Swiss origins. began in Switzerland with a schism in the Anabaptist group, or baptized again, and it was led by Jacob Amann, and basically the people who followed him became known as the Amish. They're known for simple living, plain dress, reluctance to adopt many of the modern technologies, and I found this out when I was researching them. If you research Amish, nothing comes up that they put out, whereas the Hutterite has this beautiful website showing everything that they do. I think they're trying to recruit, but it's a very nice website. So the Amish did not have anything other than what other people put out for them. So they have traditional farming. Again, they live on single family dairy farms, and in researching this, the barns are really close to the house, and the children are frequently in and out. They say the homes of both of these populations are actually very clean, but there's different endotoxin levels, and we'll talk about that. And they use horses for both field work and for transportation. So the Hutterites, and I didn't know a lot about them before, not as well known as the Amish, but it's a communal branch of the same Anabaptist group. And like the Amish, trace their roots to the Radical Reformation in the 16th century. They originated from Austrian province of Tyrol, which is very close to Switzerland, so same area geographically for the most part. They were under the leadership of Jacob Hutter and migrated to the U.S. and Canada in the late 1800s. Again, most of them live in either Western Canada or the upper Great Plains in the U.S. And they practice a very different model where there's a community of goods and they're highly industrialized. They own large tracts of land and they can afford top-of-the-line farm equipment, which they use. And this distances the young Hutterite children from the constant daily exposure to farm animals. So, again, that's really the only major difference, other than they wear flashy clothes, as you can see here. And they mention that in their website. So, the way they looked at this is they looked at environmental exposures, genetic ancestry, and immune profiles among 60 children, 30 Amish and 30 Hutterite. They looked at levels of allergens in the home, which were surprisingly low, we'll go over. And they looked at endotoxin levels in the home as well. They got whole blood from these subjects and looked at serum IgE, cytokine responses, gene expression, and PBL phenotyping and PBL levels. They also did a very unique study with a marine asthma model, which measured the effects of exposure to these dust extract from both the Amish and Hutterite homes, which is, I think, a very well done part of the study. We'll go over that. So they collected dust, where they took this extract and made a preparation for their studies of mice. They used electrostatic dust collectors, which were placed in one bedroom and living room. And they analyzed endotoxin and allergen levels. Again, it was just one bedroom and one living room. And just to say, they only did this in 10 homes of each of the Hutterite and the Amish. So they didn't do them in all of the different subjects' homes. Vacuum was also used to collect dust. So they looked at mouse model of asthma sensitized to ovalbumin. And they instilled 50 microliters of these house dust extracts, both from the Amish and the Hutterite, intranasally every 2 to 3 days for a total of 14 times, and they, gave them out ovalbumin. And then they also looked at a different model, looking at wild-type mice where they exposed these house dust extracts from Amish, both in wild-type. They also did it in mice deficient in MID88, so knocking out a component of their innate immune system. And then they did it both in MID88 and TRIF, which involves a lot more of the toll-like receptor signaling, so you knock out more of their ability to have the innate response. So there are 30 Amish and 30 Hutterites, as we discussed. The ages were 7 to 14, and they were similar with regard to age, sex, and citizenships. The prevalence of asthma in this group, in the Amish, there were 0 that had it, or were formally diagnosed with asthma, and the Hutterite, there were 6, or 20%. So in a lot of ways similar to what previous studies had shown. The total IgE level was significantly elevated in the Hutterite versus the Amish, and they also had more specific IgE to aeroallergens in the Hutterite. So this is what Teal was discussing and how similar they are genetically. So basically, they did a principal component analysis, and I don't completely understand how they came up with this graph, but basically they're looking at allele frequencies using genome-wide SNPs, and they compared Amish, Hutterite, Basque, French, a lot of different populations. And as you can see, they're very similar genetically. They actually overlap. The Amish are in blue, and the Hutterite are in red, and you can see they almost cover up because they're so similar genetically as opposed to Sardinia and a lot of the other populations in Europe. So they looked at common allergens. This is somewhat surprising. In those 10 homes that they sampled, they looked for allergens to cats, dogs, house dust mite, cockroach. And they were detectable in 4 out of the 10 in the Amish and 1 out of 10 in Hutterite. And that makes some sense for the cats and dogs since they shun pets in the house. But I was just surprised at how low that was. I didn't see that they analyzed horse, which I thought would have been interesting as well. But endotoxin levels were significantly different. The Amish had much higher levels of endotoxin, 4,399 endotoxin units, or EU units per meter squared. And the Hutterite were 648. So the Amish had a much higher level of endotoxin. They also looked at specific microbes in the mattress. And I don't want to harp on this too much, but all of these microbes are totally different. So there's a totally different microenvironment that they're exposed to in regards to these different bacteria.
Drew, did they have different kind of mattresses, or are they basically the same kind of mattresses?
Good question. I don't know. I assume they're relatively similar, but that's a good
Probably
question.
not sleep number beds or anything.
Yeah, I don't know if they shop at Mattress Depot or whatever. But yeah, it's a good question. So they looked at PBLs and found that in the Amish population, you can see here the neutrophils are much higher than the Hutterite. The eosinophils, on the other hand, are much higher in the eosinophil or in the Amish compared, the Hutterite as opposed to the Amish. So Amish have a much higher level of neutrophils and much lower levels of eosinophils, and there's really no difference in monocytes. When they looked at phenotyping of neutrophils from Amish, they looked at CXCR4 and the adhesion molecules CD11 and CD11C. And basically that looks to see whether they're recently emigrated from the bone marrow. So in other words, the neutrophils from the Amish had recently been sent out into the periphery, likely an innate immune response. They also looked at levels of HLA-DR4 and the inhibitory molecule ILT3 and in monocytes. And this basically indicates that these monocytes, while they're similar, have more of a suppressive phenotype in the Amish as compared to the Hutterite. Interestingly, there were no difference in the percentage of Tregs in these patients either, so it looks to be more of an innate immune difference, at least initially. So they looked at gene expression profiles, and this is some heavy-duty stuff, but they looked at genes that were upregulated in the Amish children and genes that were upregulated in the Hutterite. And they were organized into co-expression modules, and again, this is somewhat over my head, but they looked at whole genome co-expression network analysis, basically looking for patterns. And 18 of the genes in one module were overexpressed in the Amish, clustered in a network that were involved in TNF and interferon regulatory factor 7, 2 key proteins that are involved in the innate immune response. This I thought was really interesting data. So this gets into the mouse models that they had come up with. Basically, taking these mice that had been exposed to ovalbumin, the asthma model, and exposing them either to the Hutterite, dust extract, or the Amish. And as you can see, on the x-axis, here's the challenge, and on the y-axis, there's airway resistance. And in the Hutterite here, there's much more airway resistance consistent with the ova exposed. And in the Amish, there's a decrease in airway resistance. So they think that giving them, this house dust extract, whether that's allergens or endotoxins, can help prevent, the expression of asthma. They also looked, and also to mention, they looked at BAL in these mice and found that eosinophils in the Amish exposed mice were significantly decreased. They also looked at wild-type mice, and again, you look here at the ova Amish and saline, and they're similar, meaning when you expose these wild-type mice to the Amish house dust extract, you don't get the same airway resistance. Next, they took a Mighty 88 knockout model, again, knocking out one component of the innate immune system. When they did that, there was an increase in airway resistance in the Amish ova model. When they knocked out another component, when they knocked out MID88 and TRIF, which is a different mediator involved in toll-like receptor signaling, the effect was totally gone. So, in other words, if you knock out the innate immune function, then the Amish dust extract loses its effect. Again, the study revealed marked differences in the prevalence of asthma, despite very similar genetic ancestries and lifestyles. Again, they're very similar other than this one major difference, which is the Amish practice traditional farming, where they're exposed to environment rich in microbes. They have an increased endotoxin, documented in Amish homes. And the Amish had very distinct immune profiles when they looked at them. They had lower IgE, lower eosinophils, and higher neutrophils that has recently emigrated from the bone marrow. And the gene expression was different as well, and genes were upregulated, known to be involved in the innate immune response in these Amish-exposed individuals. Eosinophilia in the mouse model was decreased if they were given Amish dust extract, and airway hyperresponsiveness was also decreased in these mice. And there was an absence of protection, again, when they knocked out the innate immune response. So going to this innate immune response, this has evolved to sense the environment and transduce signals that calibrate the adaptive immune responses. Proteins MID88 and TRIF are involved in multiple toll-like receptor signaling. As you can see here, MID88, TRIF, involved in multiple aspects. And deletion of these molecules serves to decrease the innate immune responses and also dysregulates adaptive immunity. The fact that the loss of protection was more marked in mice deficient in MID88 and TRIF than mice only deficient in MID88 points to the fact that there may be involvement of multiple different innate pathways in these mice. And the findings suggest that an intense and sustained exposure to microbes can activate the innate pathways and shape downstream effects, including the adaptive immune system. So again, despite similar genetic ancestries and lifestyle of the Amish and Hutterite children, the prevalence of asthma and allergic sensitization was much higher. So there's not a lot of variables other than that, the fact that their farming practices are very different and they have different exposures to the endotoxin and the animals that they're frequently around. The study revealed the endotoxin level in the Amish was 6.8 times as high. There were profound differences in the proportions, phenotypes, functions of the innate immune cells. And in a mouse model of experimental asthma, the intranasal installation of house dust extract from Amish, but not Hutterite, inhibited the airway hyperreactivity and eosinophilia that they were seeing. So, just to kind of end, I want to say a big "I told you so" to someone in this room who's going to be nameless. This person used to yell at me for not cleaning my room and wouldn't let me do it. I didn't have any pets that I wanted.
Your mother's not here.
I was simply trying to increase endotoxin levels and decrease my risk of atopic disease. So,
Is that why you wet your bed?
So, Drew, did they make any suggestions about how those of us in Western society should change our lives to?
We should all become Amish. Is what they said. No, they didn't actually. They said that's up for a future study. That's a good question.
I mean, what are we going to do now that we live a certain way?
So just for a discussion, what do you think the mechanism was here? So there's, did other people have thoughts about what they're doing to the mice when they're giving them, or to the kids?
As far as when they're giving them the...
So one of the things about it is that I think this study shows, and they didn't really look at it so much in detail, but this could really reflect a shift in the gut microbiome, right? Because when you're giving them mice the intranasal dust extract, presumably they're getting a lot of that in their gut. And the same thing is probably true of the the kids in the home. They're partly probably changing their gut microbiome more so than changing their, which is a whole kind of other thing. So,
and presumably these kids are getting less antibiotics as well, which I think may be a risk factor as well, although I don't know the difference
Exactly.
between Hutterite and Amish. Actually, when I was in St. Louis, I took care of a few Amish patients, and so they
Do they ever intermarry?
Sorry,
Do they ever intermarry?
they're reproductively isolated. I don't know if they were.
You mean
what
the
happens?
Hutterites and the Amish?
Yeah, what happens
Oh, no,
if,
between the two,
if
no, they're
you
totally
remove
separate.
one from his environment and move him to a different environment? Would he then develop a different profile?
Well, each of
Speculation.
them only reproduce within their group. So
Yeah.
that's a good question.
I was wondering of another fact. When I read that article, the Amish have really low food allergy prevalence. So about a year ago, they had a big article in the New York Times where they interviewed the researcher that went there, and then he was talking about how the pregnant women are milking cows right till they go into labor. And so I was wondering if the Hutterites were different, since
They
they're
don't milk their.
because you have an intense
They don't
prenatal
milk their cows.
exposure.
They have the traditional pump system. It's very advanced. It's like any other major farming system. So I would be shocked if they were out milking their cows as well, just from their website.
Yeah. But one of the big things is that you showed the bacterial composition of the dust is completely different.
It
right?
is.
So it's
There
not
wasn't
just
one organism that was the same as far as the most prevalent.
yeah. It's not just like the total endotoxin level, kind of a generic measure of bacteria, but that actually the types of bacteria are really markedly different. So, which has been the thing that's been shown. Antibiotic exposure early on in life is associated with asthma. Method of birth and all these other things like that presumably are changing the gut microbiome. And you can show that very easily in mice that treating them with antibiotics will make them more susceptible to a asthma model like that. Interestingly enough, in that paper, forgetting who the primary author is, they actually needed to have prebiotics as well, so they had to give them soluble fruit fiber as well as the change in their gut microbiome to make them less susceptible to asthma. It's in Nature Immunology.
Well, I thought the storyline here, though, is in humans, forget the animal part of it, is endotoxin, just generic endotoxin, changes your innate immune response, and that then changes your adaptive immune response to make you less atopic. And if they weren't focusing at all on the gut microbiome as a mechanism, you're just proposing there's an alternative explanation?
Yeah, well, I think the literature, right. It does show that, like the Von Butis paper showed many years ago, that overall exposure to endotoxin was associated with protection. I guess I'm just sort of speculating on what the actual mechanism might be.
They speculated here that there was like the neutrophil data and the activation markers. It was like the Amish kids were having low-grade inflammation all the time.
Constantly, coming from the
Pro-inflammatory in this innate response was what was. You had this kind of lingering subliminal inflammation going on all the time that was knocking down the ATP.
So coming back to the way we live now, like the best thing comes back to the idea of having dogs in the house, bringing in their dirt and their endotoxin would be a practical way to mimic this lifestyle.
Yeah.
My dog eats poop and he has no allergies.
Get a cow.
Did you find it odd that when you looked at all the bacteria in their dust, that there was absolutely nothing the same between the two populations? I would think that's statistically unlikely in and of itself, with 15
It is
micrograms.
interesting, and they are in different parts of the country. So they are geographically different as well. Yeah. I didn't know what to make of that. And that's the thing, that was in the supplementary. They didn't really comment on that at all in the paper. So I don't think they had a lot of idea as to why that was the case.
I remember when this study was done in German farms country about 20 years ago, and they came up with the same findings. That if you sleep in the barn with your animals, you don't get asthma. Right. And if you sleep in the house, you get asthma. And that was quite controversial back then. That was probably 20 years ago.
2002 was
You know, it raises a question, too. If you change your lifestyle, like at age 20, let's say you have high endotoxin your whole life, and then you go to low endotoxin, are you at increased risk after you have developed your immune system of a switch over to
I agree. It'd also be
atopic
interesting
state?
to study a younger patient population when the immune system is evolving and look at a lot of these phenotypes and different cell levels.
How old were these kids again at the time of
7 to 14.
But, I mean, you would assume that the key thing is they lived in this lifestyle from day one.
Right.
That, I mean, the formative years, my gut feeling, Gary, would be once you evolved your immune system, it's done. And the only way we know to reverse atopic disease is to desensitize people. You can't, I don't think, my guess is the environment wouldn't desensitize you once you already evolved your immune system.
Yeah. Although you may have a more inflammatory response, you know, you might have more neutrophils and all of that, and that might change your overall, response to allergens, etc. But you're right. I agree with you. I think it must be something early in life or prenatal, natal, that time.
All right.
Well, if you're going to compare me to Drew, I'm obviously the Amish version of this, and he is the Everett version. He understands computers and makes nice slides and everything. So I was trying to figure out what to prevent, because usually it's something esoteric, and I want patients to let me do it. So in one day in my office, one lady comes in with this National Geographic about the DNA revolution and says, "Can you cure my son CVID with this?" And this is like a month ago. She hands me this. I'm like, I don't even know what you're talking about. And then the same day, another lady comes into my office, and she comes in and she's sitting there saying, "I don't want to get a CAT scan on my child because it's going to cause cancer." And I'm sitting there thinking, okay, so these are some of the more controversial questions we have to deal with on a day-to-day basis. So I figured maybe everybody else is struggling with the same discussion with patients. So I thought I'd review the literature because I was interested in it. So if you look at this first article, The DNA Revolution, is published in National Geographic August of this year. And the question is, that we now have the ability to quickly alter the code of life, has given us unprecedented power over the natural world. The question is, should we use it? And the controversy stems back like it was in the 1940s when we developed atomic weapons. And all of a sudden we have this ability to destroy ourselves completely. How are we going to regulate this technology? And the same arguments are coming up about this. So we have this technique called CRISPR-Cas9, which I actually had absolutely no idea what that was until a couple of weeks ago. And it stands for Clustered Regularly Interspaced Short Palindromic Repeat Units. And then you can combine that with a Cas9 enzyme, and you can actually send that into a DNA code, and you can cut out whatever gene you want. And you can insert whatever gene you want at the same place. And they can exactly place these genes exactly in the genome where you want to put that. And the accuracy of this thing is supposedly unbelievably accurate. So you can literally project where you're going to put the gene. So they've used it in some animal models to correct genetic flaws that are responsible for the diseases muscular dystrophy, CF, and one form of hepatitis. And they're actually trying to use this to remove the HIV DNA from human cells that are infected. But so far that's not been successful. So one of the things that they're looking for as a way to help us is that pig organs are evidently the organ transplant of choice from animals to humans because the pig size are similar to ours, and I guess we're more similar to pigs than any other animal. But they're trying to get rid of these viruses, and there's a group of viruses, retroviruses in pigs that are almost innate into all the pigs, and they're like the HIV virus in humans. So what they have to do is they have to get those viruses out of the pig before they can use any of these organs to transplant. And then they're looking at ways that ecologists to protect endangered species. So you can actually take DNA from a near extinct animal and you can put it in another animal of the same species and increase the variance in their DNA and hopefully increase their survivability. The plant biologists are all excited about being able to put in genes and delete genes that attract pests, and hopefully they can make these crops more immune to degradation by plants or by insects and other ways of destroying the crops without using pesticides. And basically, there's no scientific discovery in the past century that holds more promise, and none that have sparked controversy since atomic weapons of this level of controversy. So Dr. Eric Lander was the director of the Broad Institute at Harvard and MIT, and I don't know what that is, but I assume it's important because they made him the head of the Human Genome Project. And he says that after reviewing this literature, he says scientists don't have the answer to all the questions that are going to come because of this new technique that's available. And he says he's not sure who should make this decision because we can literally change human DNA any way we want right now if you're sort of in a position that it can support the research to do that. So the first part of this is an enzyme called Cas9, which basically functions as a DNA scalpel. And bacteria have evolved this system to sever and disarm the genetic code of invading viruses. So this is a bacterial defense mechanism that has now been identified in human cells as well. And it works only on a very specific target programmed into this CRISPR-Cas9 package. So the Cas9 splits the DNA, and then the CRISPR package puts in whatever DNA you want. So you actually send this thing into the genome, and it clips out what you want and adds what you want. So the CRISPR part is an RNA guide that leads the enzymes to the precise nucleotides that the complex has been programmed to cut. Again, the accuracy is uncanny, so scientists can send a replacement part to any location in the genome. When it reaches the exact place in the genome it's programmed to target, the Cas9 enzyme snips out the unwarranted DNA sequence, and to patch the break, the cell then inserts the chain of nucleosides that was delivered in the CRISPR package. So you basically send in the part you want, takes out the part you don't want, and you have annealed DNA that's totally ready for reproduction at that point. The third component is called the gene drive, and I'm not exactly sure what this is because I tried to understand it, but there's parts of the genome that can sort of override rules of gene inheritance, and they refer to these as selfish genes, and that's because they have a much better than 50% chance of being inherited in contrast to the usual 50-50 inheritance pattern in the normal gene assortment. So by attaching this CRISPR to a gene drive, then you basically can put in the desired DNA sequence, and it becomes the favored gene during natural mating. So if you put this in one host, it goes to the other one more than 50% of the time, and then you can select out for the trait you're looking for. So in one PNAS paper, this has been used in mosquitoes to try and get rid of malaria. So one of the researchers actually engineered a version of the Anopheles mosquito that they can't spread the malaria parasite, and all other biologic functions of the mosquito are intact except for this ability to transmit malaria. So this guy said he's been laboring in obscurity for 30 years, and now his phone's ringing off the wall when people read this. Can you really do this? And they're using a gene drive to make the mosquito sterile. So that's another way to end the problem. Because right now, the only way to get rid of our current virus issues with the new viral issues with small brains and small skulls is that they have to kill all the mosquitoes. But to do that, they have to blanket the entire area with pesticide. Because in order to kill the mosquitoes, you actually have to touch them with the insecticide, which means the ones that are in hiding, the ones that are indoors, you literally have to have huge quantities of insecticide to stop this. So if they can make the mosquitoes sterile, then when they breed, they'll basically breed themselves out of existence. And that's what they're trying to approach now. And then other researchers are saying, hey, if we're going to get rid of this gene, to get rid of it, why don't we get rid of all their abilities to transport diseases and put that into their genome and let them breed that into the population? So there's already arguments being described on how to even use this stuff, and we haven't even figured it all out yet, but they're arguing about what uses it has. So what's interesting is while everybody's happy with medicines that are made through genetic engineering, everybody's accepted that really well, but yet genetically modified foods and crops have met with this horrible restraint. People don't want to use it, they don't want to eat them, they think they're bad. So despite having lots of literature saying these genetically modified foods are safe, most of the public thinks that they're horrible, they won't eat them, so they can't sell them. So the only place these genetically modified foods are being used is in countries where they're starving, and they'll take whatever they can. So they're using this in lots of places in the world, but people in the United States won't buy these foods. So the companies are not even pushing the technology because they said they can't sell the product. So what they're hoping is with this technique, instead of putting in DNA from another plant into taking the corn molecule and putting it into your rice, they're saying what they're hoping is that we can just genetically modify the DNA of the native plant, that maybe people will accept that more than they'll accept genetically, quote, modified foods. And I think that's sort of smoke and mirrors myself, but it probably will work because most of us don't know the difference. And the Chinese have started taking this to a whole different level, and they have the Chinese Regenerative Medicine Institute. And they're already doing all these CRISPR studies with animals, and they're trying to modify pig corneas to transplant them into humans, and they think they've done that now. Now if they can take out these retroviruses, these porcine endogenous retroviruses, which are similar to the HIV virus, they can start using pigs to clone organs for humans, basically. And the only thing that they have to work out now is how to get these viruses out, because they think they have the other things stabilized. They tried to take non-viable human embryos and try to correct the genetic defects and see if they could raise them with cells that no longer had the genetic defect. So as opposed to the United States, the Chinese are already starting to study human cells instead of animals, so they're sort of in the next level already. And then if they mix, when they get the viruses, these porcine viruses out of the pigs, and they culture human cells in with these pig cells that have had the viruses removed, right now they can already prove that none of the DNA-edited pig cells, none of the human cells have gotten infected. So they figure that they've gotten out all this porcine endogenous retrovirus material, and they think they're ready to go to the next step, which is making organs for human transplantation. So this is also being touted by the environmentalists as a way to save endangered species. And the one that they have done the most work on is a black-footed ferret. And for any of you who have one, you're very rare animals. But all the ferrets in the United States have come from one of 7 ancestors that lived on a cattle ranch in Wyoming in 1981. And the food source for ferrets is evidently prairie dogs, but the prairie dogs have all gotten infected with plague. Then they die. Then the ferrets eat the gophers, and then they die. So basically, there's only one group of ferrets in existence in the United States. And they have some concerns that with no genetic diversity, they're basically intermarrying amongst themselves, that they're going to select out for all these negative environmental issues, and they're going to die. So what they have is a San Diego frozen zoo, and some of you probably know more about this than I do, but there's a group in California who are taking genetic material from every species that they think is going to go extinct in the next few years, and they're freezing it, so they have genetic material from all these animals. It's sort of a Noah's Ark 2016 issue here. And what they're doing is they're taking out some of the frozen DNA from ferrets that died 30 years ago, and they're going to intersperse that with the DNA of these few remaining animals in Wyoming, and they're going to see if they can increase the genetic diversity enough to let these animals survive and breed. So it's got some interesting sort of alternative uses already, and the environmentalists are thinking this is the best way to save these endangered species. So the concern over this is no discovery in the past century holds more promise or raises more troubling ethical questions than this CRISPR technology. And literally, we have the technology now to change humans, to change any animal into basically whatever you want to put in there. And the question is, how do we regulate that, and who should be the responsible parties? So I figured with this group of super intelligent people, we might be able to solve that question and pass it on. So, and I'm sure there's people in here who know more about CRISPR than I do, but it was just a fascinating thing to read that this is already mainline. It's not something that's Star Wars anymore. We can take genes out, put genes in, doesn't harm the species, and it's transferred on as genetic material native to that animal from that time forward. So, just something for you to think about when your patients ask, can you cure my kid's CVID? Yeah, Nick.
So this is all working with germ cells, is that correct?
It can work in any cell.
How can you do it with a somatic cell and then have the species pass that on?
Well, basically, if you change the gene in a gamete, then that gene becomes a preferred gene, and then it's
Right,
just
in a
passed
gamete,
on.
right, that's why germ cell.
Yeah, so basically, after the fact, you can't cure the genetic diseases, but they're already showing that if you take non-viable fetuses, which is what the Chinese are using to explain their human research, that you can actually take these genes out. And they've not been able to bring the animals back from dead yet, but they think they can take the genetic material out of them. So that's what they're using to avoid international scrutiny, is they're using non-viable fetuses to see if they can screen out the genetic defects.
Well, David Hagen gave us a talk. I don't know if David is listening in Tel Aviv right now, but about, what was it, 2 years ago, and he talked about this in the context of immunodeficiency diseases, which clearly in our field would goes back to your original question. cure my son's CVID. So what's the answer to that question?
The answer looks like it's yes.
Well, I didn't get a clear answer between the two of you.
Yeah.
Did you do this only in somatic cells, or you do it in the reproductive cells so you can then pass the corrected
The
trait
next
on?
generation.
Well, I mean, to the degree that you're doing transplants for immunodeficiency these days, I mean, I suppose that would allow you to do an autologous transplant with a corrected stem cells. I don't know.
That's currently being done here with both with Scott Aldrich and X-linked A-gamma-globulinemia. But they're not using CRISPR-Cas9 right now. They're still using a lentivirus to correct the genetic defect. But you could use this as well.
CVID may be a little more difficult since most of the cases still, Matt, correct me if I'm wrong, we don't know the genetic defect.
Right.
Right.
Yeah, that's true.
But other
But I mean,
deficiencies.
for an immune deficiency, you couldn't do this with most other genetic deficiencies where you're right. I mean, the somatic effects are so overwhelming, but where what you really need to replace is the bone marrow, you can effectively do a bone marrow transplant with an autologous corrective cell. So that's what Andy Scherenberg and Dave Grossling and those guys are doing.
Another thing they're trying to do is they're trying to genetically edit out the HIV DNA from cells that are infected, and they're thinking if they can do that in a host, they could get enough of the enzyme in there that might be able to genetically alter the infected cells in humans with the disease.
Yeah. I know one problem they've had with this. I mean, this sounds great. The actual efficiency of this system is not outstanding, and it's particularly not great currently in T cells. So you, from what presentations I've seen, you get like a 10 to 20% effective rate in T cells. So where you're thinking about trying to eradicate, for example, HIV from T cells, that would be a challenge. But it will certainly get better, I'm sure.
It's interesting how people accept genetic modification. That means getting insulin for free out of mice and rats, but then they don't want to eat genetically modified corn or genetically modified fish because they're afraid of them.
It's true, although, I mean, despite the fact that people don't like it, it's overwhelming in our food chain, the amount of genetically modified stuff.
The Europeans have rejected it much more than the U.S. I don't think it's been that rejected in U.S. population.
Yeah.
The malaria thing I found intriguing, and with the Gates Foundation here, do we know if this is going on here? I mean, that seems like a brilliant way to try and solve the problem with malaria as opposed to tents and netting and insecticides.
Well, they didn't comment specifically anything about the Gates Foundation. This was more of a National Geographic article as opposed to a scientific one. But the guy who's the mosquito expert, I didn't realize there's like 500 different kinds of mosquitoes. And what they're really working on is a way to make the mosquitoes all sterile, so that you release them into the population, they all die and they can't reproduce. So instead of insecticides, they said a couple generations of these sterile insects, and then the breed is going to basically disappear.
Right.
So then we're like, are we altering DNA? Are we going to do something good? What do mosquitoes do that's good for us? We won't find out till we kill them all, and then we won't have malaria, but we'll have some other horrible disease that comes
Yeah, well,
in.
what happens if you wipe out a total species in the environment? Who eats that species? And they're affected. I mean, changing the...
That's the cataclysmic response.
Yeah.
And then when you look at the pollution that we're putting into our country, now they can measure like birth control levels of hormones in orca whales. I mean, where does that come from?
Yeah.
It's obviously just coming from contamination that we're doing to ourselves. And you just wonder, I keep wondering back to the old Star Trek, the Prime Directive, is, you don't alter anything because you're going to screw something up.
Right.
And we're getting into that ability now that we can screw up a lot of stuff. And boy, the controversy with this technique, it's like religion and politics. There's people that are just polarized on each side. And they all cite a lot of great data, but nobody's really agreeing on anything yet, other than the technology is impressive. Well, that's enough of that confusion. So now we're going to another level of confusion, which is the radiation safety of CT scans. And this is, I don't know how many of you deal with a lot of CTs in kids, but the pediatricians in my clinic, when I want to CAT scan a kid, I mean, they want to take out my spleen. They're just really against this altogether. And the alternatives are really not that great, but these people are convinced that a CT scan is going to give their kid cancer and they're going to have problems. And I've just struggled with this concept forever, and then when these two people came in back to back in my office, I said, okay, this is what I'm going to do. So there's many articles in the press and scientific literature over the past years that says CT scan may cause cancer, and that physicians should be cautious with their use. So although there's broad agreement that CTs may cause cancer, there's still controversy surrounding the question is, do they? And is it really possible? So radiation doses are indeed higher with a CAT scan than conventional imaging, but they're still 10 to 100 times lower than the dose that we've experienced in the past to increase the risk of cancer. So despite the fact that these doses are far below those known to cause cancer, the magnitude of the concern among patients and medical professionals is still unreasonably high. And if you get into a discussion with a parent about CT scanning a child, it gets ugly really fast. And MRI scanning a child, I think, is just cruel because you got to knock them out. They're in a scanner for an hour. It's just a huge difference. So it's definitely the best technology. And one of the things I heard about Obamacare is what President Obama thinks is one of the failings of our medical system is that we CT scan too many people. And that if we just quit CT scanning people, all of our economic woes would go away from medicine. But I'm not quite sure it's that simple. So the number of scans in the U.S. is over 81 million in 2014. And then the New York Times published an article from two physicians that said that CT scans in our country and other countries are killing people. And you know, the New York Times had a readership of about 5 million people, and it's like, now we're killing people with our x-rays, and that didn't go over well with the public. So they wanted to get, some realistic data, and there's a trend now that people who really need CAT scans who won't do them because they'd rather not have a CAT scan than the risk of the radiation, and the risk is incredibly low. So a lot of doctors won't order CAT scans because they don't want to argue with the family and the patients about why it's appropriate to do that. So then you're stuck with inadequate imaging like an ultrasound when you should do an MRI or a CT. So there's different ways to quantify radiation doses, and there's the CT dose index, which is reported in milligrays, and energy from X-rays is described in milligrays, which is an exposure, and millisieverts, which is a dose. So when you look at radiation levels, what radiation is put out by the machine is not necessarily the dose that the patient gets into them. So you have to figure out ways of estimating total radiation dose. So there's specific dose estimates, which is SSDE, and there's a CT dose index, which is, there are two different ways to basically measure what the potential radiation dose of a patient is, because there's no way for us to actually measure what the radiation dose is. So for a head CT, the potential exposure is 50 to 60 milligrays, and a gray is 1 joule per meter squared, and that's an energy unit, and that's how they sort of quantify all these things. So if you look at a colon, it's 50 to 60 milligrays, and if you look at an abdomen and pelvis, it's 15 to 20, and if you look at a head CT, it's like 4. And if you look at regular conventional imaging, it's like 0.01. So there's very little energy done with conventional X-rays. And now the question is, what are these doses doing to people? And for a comparative guide, they have, from cosmic rays, the average person gets about 3 millisieverts per year, unless they're exposed to radon. So they separate radon out because that's a very local phenomenon. So if you look at conventional radiography, take a hand film, it's 0.01 millisieverts. A dental bite wing is 0.01, chest X-ray is 0.02, and a mammogram is 0.02. Whereas a head CT is 2, a chest CT is 7, abdomen CT is 8. So most imaging is between 1 and 10 millisieverts. And then nuclear imaging, for a lung scan it's 2, a bone scan is 4, and a cardiac imaging is 14. So that's just sort of reference numbers for you to think about. So if you look at a whole body dose of radiation, most of our long-term risk data comes from people that got one single large dose of radiation. And a lot of this is from the Atomic Bomb Casualty Commission, and they're still following these people from Hiroshima and Nagasaki. And they noticed an increase in leukemia as early as 2 years after doses in excess of 200 millisieverts, although sometimes this doesn't appear for 55 years. So you got to ask yourself, if lots happened in 55 years, can you really track it back that far? For solid organ tumors, the latency period is 10 to 40 years. So there's ongoing epidemiologic studies from the Atomic Bomb Casualty Commission that found an increase in cancer mortality at doses above 150 milligrays. So that seems to be the safe dose upper limits. And then if you do the whole body doses below 100 to 150 milligrams, they've not shown any increase in cancer. So that's the data that's most often cited. So if you look at other studies where you look at people that live in high altitude, like in Telluride, Colorado, you may have elevated background radiation, you get 100 to 250 millisieverts per year, because the higher you go, the less the atmosphere shields you from cosmic exposure. And in the UK, they have people that have very large exposures. Most of them are healthcare workers, and they'll have up to 200 to 500 millisieverts per year. But what they're looking at is episodic dosing doesn't seem to have the same effect as a single large dose. So these people that get 200 to 500 millisieverts per year, you follow them for 30 years, and they don't have an increase in cancer. Whereas if you look at the bomb survivors, and they had this huge single dose, that statistic seems to come through very quickly. And then they had 64,000 Canadian TB patients that had repeated chest fluoroscopies over years and years, and they were able to add up their doses of radiation that was basically equal to the people who survived the atomic blast. And the numbers there, people that got more than 200 millisieverts after the blast, they didn't survive, and under 200, they did survive. So they're finding that there's no correlation between risk and dose. So all the studies are still controversial as to which is the answer. Is a cumulative dose the same as a fixed single dose? And what are the actual risks? So if you look at all the different studies that focus on different types of exposures in different populations, using different methods of estimating dose, there's no convincing evidence of any increased cancer at doses less than 100 millisieverts. So I think the bottom line we can tell patients is if you get less than this dose in a year, you're not going to have any increase in risk of anything. There's 2 studies that are ongoing in the people that have had multiple CTs in childhood. And a lot of these are spina bifida kids and a lot of these skeletal abnormalities. These kids are followed with x-rays and CTs all the time. So they're actually following them prospectively now that they've been able to calculate all the dose of radiation they've had. And that's an ongoing study that's going to go for about another 15 years to see if they can answer the question. And then there's 2 studies that looked at CT scans in childhood may increase the risk of cancers later in life, but both of these studies, when they were analyzed by the supposed experts, they had really methodologic flaws that were quite bad, that really biased the data. So there's 2 more recent studies that address these 2, and they don't show any association, so the controversy continues. So if you look at the data in kids, in 10 years following CTs, there's a 1 in 10,000 increase in brain cancer and leukemia. If you look at the 2 studies that are done in the United States, and if you look at the 2 studies that are done in Germany and France, with 70,000 and 45,000 patients each, they showed no increased risk in kids that had that radiation dose. So there's something that's not quite equal between the doses, and the data continues to be really murky as to what that means. So even though the data is equivocal on radiation risk in children, there's a consensus that the lowest dose possible is always the dose we should use. And I think that's the ongoing concerns. But despite all the questions that have been raised about a link between cancer and CT scans, there's not a significant convincing case for a causal link yet at rates under 100 millisieverts. So I think you have a bottom line that you can tell patients, you know what the CAT scan is going to give them in terms of radiation. So the current wisdom is that you should be able to tell people you're not at any risk at all. Whether they'll believe you or not, I'm not sure. But the data is very clear. Under 100 millisieverts, there is no link with cancer, and that's per year. So if they get 90 millisieverts for 10 years in a row, it looks like that's okay. Anyway, this was just something that I find difficult, so I thought maybe you had the same question. Nick?
Was there any reference in your readings to the Hanford study that was done at the UW with the Hanford workers and increased risk of, I think it was thyroid cancer at the time? Did you see anything about that?
That wasn't specific. I didn't read that article to do this, but it wasn't referenced in this article either. Most of this was looking indirectly at kids. That was really the concern. They were looking at the adult data and trying to extrapolate that back to children, so I'm not sure that would have helped. But the intermittent exposure is clearly different than a single dose.
So how does a one-time CT compare to your risk, your exposure just from cosmic exposure?
Well, you get 3 millisieverts per year in a cosmic exposure, and depending on what organ you CT, if you get CT scan the head, that's equivalent to 2 and 2.3 years of cosmic exposure. Unless you live in Telluride, Colorado, which is a lot more. So.
Well, certainly diverse topics today. always stimulating conversation with this group. Next week, don't we have a visitor from St. Louis?
Yeah. Well, yeah, I grew up in Idaho, at 6,000 feet. Yeah. My picture that towards Actually, that's the same thing.
I'm glad I heard it.
Yeah, like that. Yeah, low-dose CT scans. I mean, the controversy is about that.
What's the highest that you do? The high resolution.
Well, yeah, there is how many CT scans don't really have very much radiation dose because you're blasting it through the lung.
They didn't go that far. They basically used
Mostly we use
so the highest city with any real population, you have Flagstaff, Arizona, which is 8,000 feet, and you have Telluride, which is 10,000 feet. So they figured, let's go for the highest altitude. But 100 to 250 millisieverts a year, that's like 20
well, I mean,
a year.
finally, people are
So, but they don't have enough long-term data to answer this. But it's that's a question that's sort of ongoing.
Yeah, that's good stuff. Thanks.
If your patient gives you a national geographic, that means they're looking for something. Nice.
But before that, most of the data
Matt, you get tired of my mail?
was if you're a cigarette smoker,
So, you have anything, any thoughts of how Zentra's equivalent versus IVIG? Do you still want to start on an IVIG?
No, I mean,
Yeah, can I?
Have to get another.
yeah, right, and then it depends on what your
First drive up to North.
Yeah, interesting. I'm getting a lot of these people now.
If there's nothing, I think then you can do it here. But it really is called low-dose CT scanning. So
And I was telling you, the benefits of Zentra and equivalents, you never have to. And you're
It sounds like that'd be safe because
yeah,
the shot
But the
that's the
data
worst
that
yeah, right, exactly.
versus this severely.
You want to help the take the food upstairs?
But I mean, I do think that argument that people sometimes
You'll take this upstairs.
very little evidence. Probably should.
going to the diocese.
I've always argued with my dentist